Hello everyone, welcome to Dr. K. Primer Biotech Lecture Series. In today's lecture, we will be not teaching any specific topic. Instead of that, my students have framed around 30 questions on the Agrobacterium tumefaciens and TI plasmid features. So those questions will be discussed in today's series. So these questions, 30 questions are framed by the students after listening to my class on Agrobacterium tumefaciens and TI plasmid features. So I thought these questions would help you also to understand the Agrobacterium tumefaciens and TI plasmids. Hence, I'm taking time to present those questions, and I guess you can take the advantage out of these questions. Yeah, the very first question is, which region of the TI plasmid integrates into the plant genome? Ori region, opine catabolism genes, tDNA region, and virulence genes. So TI plasmid consists of all these genes, but which region get integrated into the plant genome? So here the answer is C, tDNA region. Which Vir protein acts as a pilot protein helping tDNA enter the plant nucleus. Vir B, Vir D2, Vir C, and Vir E3. As you all know that Vir genes are responsible for transfer of tDNA into the plant cell, but here Vir D2, which has nuclear localized signal, and binding capacity to the tDNA, that's at the pi prime end. So Vir D2 acts as the pilot protein, helping the tDNA to enter into the plant nucleus. Agrobacterium tumefaciens is a gram-positive bacteria, gram-negative bacteria, actinomycetes, or cyanobacterium. It's a gram-negative bacteria. Which crops are mainly affected by crown gall disease? Rice and wheat, grapes, fruits, trees, and sugarcane and maize, tea and coffee. Mainly, they are affected by grapes and fruit trees. The fifth question, the crown gall tumor formation is mainly due to increased production of ethylene, uncontrolled cell division due to tDNA genes, nutrient deficiency, fungal co-infection. The tDNA region in the TI plasmid, that's a tumor-inducing plasmid, carries cytokinins and auxin genes. Those genes are responsible for uncontrolled cell division. The answer is B. Which virulence gene is responsible for binding of tDNA in the bacteria? So Vir A, Vir D2, and Vir E, and Vir D. So here, which virulence gene is responsible for binding of tDNA in the bacteria? So Vir E binds to the tDNA in the the tDNA is formed, single strand cDNA formed after the breakdown of a single strand breaks in the TI plasmid, and Vir E binds to the tDNA. Why is Agrobacterium naturally more effective in dicots than the monocots? Dicots produce testosterone, testosterone to activate the virigens. Dicots have thinner cell walls. Monocots lack chlorophyll. Dicots divide faster. The answer is A. Dicots produce the testosterone to activate the virigens. What is Agrobacterium tumefaciens best known for? Nitrogen fixation, causing root nodules, causing crown gall disease in plants, producing antibiotics. So, Agrobacterium tumefaciens is best known for causing crown gall disease in plants. The answer is C. Which compound activates the virgins during the plant transformation? Which compound activates virgins during plant transformation? Auxins, testosterones, cytokinins, and gibberellin. Answer is B, testosterone. Which of the following is a common application of Agrobacterium tumefaciens? Production of biofuels. A. Development of transgenic crops. Bioremediation of pollutants. Production of antibiotics. The answer is development of transgenic crops. So here, to develop the transgenic crops, Agrobacterium tumefaciens used as a gene transfer tool. Which system is widely used for plant transformation using Agrobacterium? Binary vector system, gene gun system, CRISPR system, transposon system. The answer is binary vector system. 12th, Agrobacterium tumefaciens is called natural genetic engineering because it causes crown gall disease. It can transfer its DNA into plants. It produces antibiotics. It fixes nitrogen. Because nature genetic engineer is Agrobacterium tumefaciens because it is able to transfer its DNA into plants and get integrated into the plant genome. Virgins involved in tumor hormone synthesis, transfer of tDNA into plant cell, open utilization, plant immunity. So, virgins are involved in the transfer of tDNA into the plant cell. 14th, which type of nucleic acid is transferred from Agrobacterium into plant cells? mRNA, rRNA, tRNA, tDNA. The answer is D, that's a tDNA. tDNA is a part of TI plasmid and that is transferred from Agrobacterium to the plant cell. Which molecules are secreted by wounded plants attract the Agrobacterium? Flavonoids, amino acids, polysaccharides, and fatty acids. Basically, here answer is not given properly, but we can say polysaccharides and testosterone. 16th, Agrobacterium tumefaciens infect mainly dicots, monocots, both dicots and monocots equally, only gymnosperms. The wild type Agrobacterium tumefaciens infects mainly dicots. The answer is A. Which gene cluster helps transfer of tDNA into the host genome? Housekeeping genes, virulence genes, open catabolism genes, and resistance genes. If you look into the TI plasmid basically have a tDNA and vir genes, open genes, and origin of replication, so on. The option B, virulent genes are responsible for transfer of tDNA into the host genome. 18th question, which part of the TI plasmid is transferred into the plant cell? Vir genes, antibiotic resistant genes, tDNA, origin of replication. As I told you that vir genes are there, tDNA is there, and open genes are there, origin of replication is there, and open catabolism genes are there, but only the tDNA region of TI plasmid is transferred from bacteria to the plant cell. So the answer is C. What is the size of TI DNA. That's a tumor-inducing DNA. 200 kb, 100 kb, 150 kb, and 320 kb. So depends on the species. It ranges from 140 to 200 kb. So roughly we can say around 200 kb. Answer is A. The virulence trait of Agrobacterium tumefaciens is born on chromosomal DNA, TI plasmid DNA, cryptic plasmid DNA, both chromosomal and plasmid DNA. The answer is B, that's a TI plasmid DNA. Because all the genes which are responsible for taking identifying the wound and nicking of the uh, tDNA, and tDNA consists of auxins and cytokines genes and opins. So TI plasmid is responsible for virulence rate of Agrobacterium tumefaciens. What is the disarmed Agrobacterium tumefaciens strain? It means A, lacking of virgins, B is missing of tDNA borders, C is divide of oncogenic genes, and D is unable to infect the plants, or E is antibiotic resistant. So missing or lacking of virgins is called as disarmed Agrobacterium tumefaciens. What is the major limitation of Agrobacterium mediated transformation? High mutation rate, A, limited host range, mainly dicots, B, and the low DNA integration efficiency, C. D is none of the above. The answer is limited host range. This is a wild type. Nowadays, Agrobacterium mediated transformation is very well established in fungi, algae, and plants, and even mammals also. 23. Virgins in TI plasmid are essential for open catabolism, tDNA transfer to plants, plant hormone production, and so here the answer is they are responsible for a transfer of tDNA into the plant cell. The 24th question, crown gall disease in plant caused by Agrobacterium tumefaciens is similar to which human disease? Bacterial toxin disease, viral fever, cancer, none of the above. Answer is cancer, because uncontrolled cell division happens, then they form a crown galls. 26. Which of the following is direct application of disarmed Agrobacterium tumefaciens in biotechnology? Nitrogen fixation in cereals, herbicide resistance crops, production of bioethanols, biodegradation of plastics. Which of the following is a direct application of disarmed Agrobacterium tumefaciens in biotechnology? So nitrogen fixation in cereals, herbicide resistance crops, and the production of bioethanol, biodegradation of plastic. The more relevant answer for this question is herbicide resistance crops. Which vir gene encodes the endonuclease that nicks the tDNA border sequence? Vir A, Vir G, Vir D2, Vir B. The answer is Vir G, which is binds to single DNA and breaks the single. It's a endonuclease, breaks the single strand breaks. 28. The genes responsible for pathogenicity in Agrobacterium tumefaciens are vir genes, lac gene, ox genes, strip genes. The answer is vir genes. What is the primary use of Agrobacterium tumefaciens in genetic engineering? DNA mapping, DNA modification, gene transfer, DNA fingerprinting. The answer is C, gene transfer. In genetic engineering, the tumor-inducing genes of TI plasmid are replaced by antibiotic resistance marker, bacterial replication genes, virulence genes, and photosynthetic genes. So here you can say, in genetic engineering, the tumor-inducing genes of TI plasmid are replaced by they are replaced by antibiotic resistance marker to select the transferments? The answer is A. Opins are secondary plant metabolites toxic to bacteria. Unusual amino acid derivatives synthesized in plant tumors. Antibiotics secreted by the plants. And the D is carbohydrates used by insects. The answer is there are unusual amino acid derivatives synthesized in the plant tumors. After formation of tumors, the tDNA integrated into the plant, then the open genes are expressed and they produce the unusual amino acid. They are used as a carbon and nitrogen sources. These are the questions framed by my students, and I'm really thankful to the students who have framed these static one questions. So this kind of exercise will make them to understand the class, and when and also they will get depth of the subject when they are making the 30 different questions from those two classes. Thank you, my dear students, and thank you, my dear subscribers, for listening carefully and getting something out of this lecture. Thank you. Hope you like this question bank series explanation. And if at all you like it, try to subscribe my channel. That's a Dr. K. PREM-PRIMER Biotech lecture. Thank you all.